Genetic drift plays a crucial role in shaping the genetic structure of populations, particularly in small, isolated groups. This phenomenon, a fundamental mechanism of evolutionary change, involves random fluctuations in allele frequencies over generations, often leading to reduced genetic variation and increased genetic differentiation among populations. In primates, especially those confined to isolated habitats, genetic drift can have profound effects on genetic diversity, adaptation, and survival. From the remote forests of Madagascar to the fragmented landscapes of the Amazon, isolated primate populations serve as natural laboratories for studying the impact of genetic drift. Through extensive field studies, genetic sequencing, and population modeling, researchers have documented the effects of genetic drift across various primate species, revealing patterns of genetic bottlenecks, founder effects, and loss of heterozygosity. By comparing different primate populations, scientists have uncovered how isolation, population size, and historical events shape genetic diversity. This documentary delves into these findings, presenting case studies from distinct ecological settings and discussing the broader implications for conservation biology, evolutionary theory, and primate survival. Genetic drift is particularly influential in small populations, where chance events can lead to dramatic shifts in allele frequencies. Unlike natural selection, which favors traits that enhance survival and reproduction, genetic drift is a stochastic process that can reduce genetic variation independent of adaptive value. In isolated primate populations, genetic drift often occurs due to founder effects, where a small group colonizes a new area, and genetic bottlenecks, which occur when populations are dramatically reduced in size due to environmental changes, habitat fragmentation, or human interference. One of the most striking examples of genetic drift in primates comes from the lemurs of Madagascar. Madagascar's primate populations have been isolated for over 60 million years, leading to extreme cases of genetic drift. The island's lemur species exhibit high levels of genetic differentiation, often with very limited gene flow between populations. Studies on the endangered II, Daubentonia madagascariensis, have shown that different populations possess unique genetic markers, suggesting that genetic drift has played a more significant role than natural selection in shaping their genetic makeup. Similarly, in South America, the fragmentation of the Amazon rainforest has led to the genetic isolation of many primate species. One notable example is the golden lion tamarin, Leontopithecus rosalia, whose population was reduced to fewer than 200 individuals in the 1970s due to deforestation. The resulting genetic bottlenecks significantly reduced genetic diversity, making the species more susceptible to diseases and environmental changes. Conservation efforts, including reintroduction programs, have attempted to mitigate these effects, but the genetic consequences of past drift events remain evident in modern populations. Founder effects occur when a small number of individuals establish a new population carrying only a subset of the genetic diversity from the original group. This phenomenon has been well documented in island-dwelling primates, where colonization events have led to unique evolutionary trajectories driven by genetic drift. The primates of the Caribbean islands provide a compelling case study. The introduced populations of vervet monkeys, Chlorocebus sabaeus in the Caribbean, particularly on St. Kitts and Nevis, originate from a small group of African ancestors brought by European traders several centuries ago. Genetic studies reveal that these island populations have significantly lower genetic diversity compared to their mainland counterparts, with certain alleles becoming fixed due to drift. Despite these genetic constraints, the vervets have adapted to their new environment, demonstrating how drift and selection interact in shaping evolutionary outcomes. Another example is the Japanese macaque, Macaca fuscata, populations inhabiting the small islands off Japan's coast. Genetic analyses indicate that these island groups have experienced severe genetic bottlenecks, leading to pronounced founder effects. The Yakushima macaques, in particular, display reduced genetic variation and distinct mitochondrial DNA haplotypes compared to mainland populations. These findings highlight how isolation and small population sizes can amplify the effects of genetic drift, leading to divergence even over relatively short evolutionary timescales. Genetic bottlenecks occur when a population experiences a sharp decline in size, leading to a loss of genetic variation. This phenomenon is particularly evident in primates affected by habitat destruction, hunting, and disease outbreaks. The consequences of genetic bottlenecks include increased homozygosity, reduced adaptive potential, and higher susceptibility to genetic disorders. One of the most well-studied cases of a genetic bottleneck in primates is the mountain gorilla, or Gorilla barangay. With fewer than 1,000 individuals remaining in the wild, this critically endangered species has experienced multiple bottlenecks due to poaching, habitat loss, and civil unrest in Central Africa. Genetic analyses reveal extremely low levels of genetic diversity, with many loci exhibiting homozygosity. The implications of this genetic drift-induced bottleneck are profound, as reduced genetic variability limits the gorilla's ability to adapt to environmental changes and resist disease. Similarly, the Bornean orangutan, or Pongo pygmaeus, has suffered dramatic population declines due to deforestation and hunting. A genetic study of Bornean orangutans found evidence of recent bottlenecks, particularly in populations living in highly fragmented forests. These populations exhibit lower genetic diversity and higher levels of inbreeding, raising concerns about their long-term viability. Conservation programs have attempted to counteract these effects through habitat restoration and genetic monitoring, but the legacy of genetic drift remains a challenge for recovery efforts. A comparative approach reveals stark differences between island and mainland primate populations regarding the effects of genetic drift. Island populations generally exhibit lower genetic diversity, higher levels of genetic differentiation, and greater fixation of alleles compared to their mainland relatives. These patterns arise due to the combined effects of founder events, small population sizes, and limited gene flow. One of the most striking comparisons comes from the study of Rhesus macaques, or macaca mulatta, in South Asia. Mainland populations of Rhesus macaques display high genetic diversity due to extensive gene flow among groups. In contrast, isolated island populations, such as those on Cayo Santiago in Puerto Rico, show significantly lower genetic variation. Established in the 1930s from a small founding population, the Cayo Santiago macaques have since experienced strong genetic drift, leading to distinct genetic profiles compared to their mainland counterparts. Despite this, the population has thrived, demonstrating that while genetic drift reduces diversity, it does not necessarily preclude survival. A similar contrast is observed in howler monkeys or alouatta species across Central and South America. Mainland howler monkey populations maintain high genetic variability due to their extensive range and connectivity. However, island populations, such as those found in the Brazilian Atlantic forest fragments, exhibit reduced genetic diversity due to isolation and habitat fragmentation. These findings reinforce the idea that genetic drift exerts a stronger influence in populations with restricted gene flow, leading to rapid genetic divergence. Understanding the role of genetic drift in isolated primate populations has significant implications for conservation strategies. Genetic monitoring can help identify populations at risk of losing genetic diversity, allowing conservationists to implement measures such as habitat corridors, translocations, and assisted gene flow. In species where genetic drift has led to severe bottlenecks, genetic rescue efforts, introducing individuals from other populations to increase diversity, may be necessary to prevent inbreeding depression. The case of the Florida bonneted bat, Eumops floridanus, though not a primate, provides an instructive parallel. Conservationists have considered introducing individuals from genetically distinct populations to counteract the effects of genetic drift and inbreeding. Similar strategies have been proposed for critically endangered primates, such as the Tapanuli orangutan, Pongo tapanuliensis, whose small population size makes it highly vulnerable to genetic drift. Future research should focus on integrating genomic data with ecological studies to better understand how genetic drift interacts with other evolutionary forces. Advances in whole genome sequencing and population modeling offer new tools for assessing genetic variation and predicting the long-term impacts of drift. By combining fieldwork with cutting-edge genetic analysis, scientists can develop more effective conservation strategies to safeguard the genetic health of isolated primate populations. Genetic drift is a powerful evolutionary force that shapes the genetic landscapes of isolated primate populations. Through case studies from Madagascar's lemurs, the Amazon's tamarins, Japan's macaques, and Africa's mountain gorillas, this documentary has highlighted the profound effects of drift in small, fragmented populations. The comparative analysis between island and mainland primates underscores how population size, isolation, and historical events contribute to genetic differentiation. As primates face increasing threats from habitat destruction and climate change, understanding the role of genetic drift becomes even more critical for conservation efforts. By leveraging genetic research, conservationists can develop strategies to mitigate the adverse effects of drift and ensure the long-term survival of these remarkable species. The study of genetic drift in isolated primates not only enriches our understanding of evolution, but also provides essential insights into preserving biodiversity in an era of rapid environmental change.